Okay, thank you so much for having me, and thank you, the organizers, to have us assembled here. I think it's going to be a really exciting program with many exciting speakers that I haven't seen before. So thank you for that. Now, the big question that fascinates me in my lab is how and why the states of others move us so much. So if you have a look at this young Ukrainian boy that just lost part of his family, if you look at his facial expression, at his tears, and at his gaping mouth, I think all of us experience distress. And the question for me is, why is it that we don't just understand that this boy is in distress, but why is it that it moves us as well? And often it moves us to the point where we really have an urge to do something for him. And why is that? Now, in this talk, I'd start by first very briefly looking at what we know from human and monkey work about what happens just while we witness the state of others. Then I'll ask whether other animals like rodents also experience this kind of emotional contagion. Then I'll try to look at whether the brain system that are involved in this emotional contagion actually contribute as well to an aim to be prosocial. And finally, I'll have a look at whether empathy, so to speak, just happens to us, or whether it is something that we can deliberately control. I'll also start the timer just to try to stay in time here. So first, what happens in our brain while we witness the state of others? So for me, that work actually started with work that I did in monkeys, where we were recording from the premotor cortex, uh, because what we saw there was that, of course, all the neurons there are involved in controlling the monkey's own actions. So, for instance, grasping a small object. But what we observed was that a lot of these neurons also responded while the monkey was simply seeing me perform similar actions, like grasping an object with my hand or with my mouth. And what was intriguing there for me was that for the first time I was realizing that while we witness what other people do, we don't just process it in our sensory cortices like the visual cortex. We don't just process it in our cognitive regions, but we actually map it onto our own body. So with neurons like that, you might think that while you see me kind of open this bottle and take a sip from it, hmm, you wouldn't just see me do it, but you might be mapping it onto bodily centers in your brain. Now, you might argue, of course, I'm not a monkey, so no, would I do the same thing? So, in humans, what we can do easily is put people in a scanner, ask them to perform certain actions, like scooping soup out of a bowl, and then we can map the brain regions involved in motor control, which we see here in red. Then we can also show you a movie of someone else performing a similar actions. And what you see are these blue regions here. And what is interesting here is that we see this partial overlap. So it's not the case that every brain region that's involved in motor control gets reactivated when you see the actions of others. That's why you don't all grasp for your bottles when you see me do it. But there is this partial overlap. And from the monkey work, we know that it's not just a blob in fMRI, but it's actually neurons that are involved in my own motor control that get reactivated when you see those of others. Now, initially, those were separate blobs in an image, or separate regions we studied in the monkey. But now, by using an ultra high field fMRI, where we can look at different layers, and ECOG recordings that we can perform in human patients, we're starting to see as well how they work together. So if instead of these isolated actions we used in the beginning, we show people more complex actions like someone cleaning their glasses or someone preparing breakfast, we see that initially the visual information goes to visual cortices and then onwards to parietal and the premotor cortices that we had studied in the monkey as well. But as soon as you can start to realize what the person is doing, we can actually measure that the predominant direction of information flow inverts Now it is actually the premotor cortex that is sending hypotheses of what the person is probably going to do next, that is sent all the way back down to visual areas where it has an inhibitory function that actually then reduces the way that you process expected regions. So what we see here is we went from neurons in the monkey to a circuit level that we can do very well in humans, to now actually starting to look at how these centers integrate to create what I call a predictive brain for for the actions of others. Now, of course, while you looked at this Syrian boy, you were probably not thinking about just looking at the actions. You were probably more thinking about sharing part of his sensations and emotions. So, how about sensations and emotions? Well, the first experiments we did were, for instance, on sensations. We simply put someone in the scanner, touch their legs. This allowed us to map the sensory sensations in the brain when it's on your own body. And then we showed them images, very exciting little movies of legs being touched. Don't laugh, please. Those are my legs, so it's not funny. And there we saw that the area that discriminated most, the site of touch versus non-touch, was this blue region here. And again, there was this overlap where basically you map the bodily sensations of others onto a substrate of your own bodily sensation. Now, how about emotions? Well, that's a little bit harder to study in the lab. I could take you, put you in the scan, and ask you to be happy for me for 10 seconds while I measure your brain activity, but that's probably not going to work very well. So, what we came up with was putting people in the scanner with an anesthesia mask on their face. And then they ask you, why do I get an anesthesia mask? But we just told them to wait a little bit. They were going to find out soon enough. Then what we started to do was to puff pleasant smells like strawberry and mint in there. They figured, gee, that's not too bad. But then we switched to butyric acid or sulforamarketan. Now that worked pretty well. We had to take one subject out because he started vomiting in the scanner. But for the other ones, we could measure brain activity while they were feeling an acute kind of visceral emotion. And what you see there was the insula was particularly involved in feeling these states. Then we showed them movies of other people undergoing similar experiences, like taking a bit of juice from a cup and looking disgusted. And what we saw there was that the anterior insula was reactivated during the observation. So again, it looked a little bit what we had seen for actions and sensations, but this time for emotions. Now, the other emotion we could study very reliably is pain, because we can just go in the scanner and slap people on their hand. And what we see there is that in addition, when they then see similar sensation, you get again part of the insula, which is therefore not very selective for disgust. It's activated by a lot of states. But you also see the cingulate cortex, which is a little bit more specific for the sufferance involved in pain. So patients that have a lesion in this part of the cingulate still know when they cut themselves, but it doesn't disturb them anymore. They lose kind of the sufferance. And we also see when you can really see what is hurting the other, we see the somatosensory cortex again being active both when you feel it and when you observe it in others. So when we started publishing these kind of findings, people went ahead pretty quickly and said, gee, so that's why we share and care the emotions of others, because our brain maps it onto our own neural substrates for these emotions. Now, the problem is that it's a nice speculation, but from neuroimaging, we don't really know whether that's true. So first of all, we need to know whether in an area like the cingulate that is activated by a lot of things, we would actually find that there's at the level of neurons, perhaps two separate populations dealing with my own emotions and those of others, and then it wouldn't really get to mix the two, or whether we would really find single neurons that map your emotion onto mine. Also, to know whether there is causality, we would really need to be preventing this activity. and seeing that you would now no longer care about the emotions of others. And that is very difficult to do for these deeper brain regions in humans. So we figure what we need here is to go back to an animal model where we can record neurons and manipulate brain activity. And therefore, we kind of, while doing that, also ask ourselves, how old is it actually to share the emotions of others? Would rats and mice already have that ability? Now, before we go to talking of empathy in rodents, I think it's important to decompose a little bit what we mean with empathy, right? Because in humans, we use it to refer to a whole kind of tree of phenomena. Then, so I think while we see this young Ukrainian boy, we can distinguish the simplest form of phenomena, which is called emotional contagion. It just means that while you see a distressed person, you start to feel distressed yourself without any necessary cognitive awareness of the fact that you're distressed for or because of the boy. Next up, we have what we would call empathy proper, which is feeling distress, but knowing that it is actually the boy's distress that you're now feeling. And finally, you could have a real prosocial tendency, which is when you stop to just feel and you actually go out to do something for the boy. And I think today we're going to see several lines of evidence to show that rodents have some of these phenomena, but we have to be aware that empathy proper is actually very difficult to measure in rodents because it's hard to ask them whether they know that they're feeling something for someone else. On the other hand, emotional contagion and prosocial behavior, I hope that we'll be able to convince you that rodents actually have these phenomena. So the experiment that we did is actually very simple. We simply put two rodents face to face, and then we give a couple of foot shocks to one of them. So, of course, the demonstrator, when they get the foot shocks, get scared, and therefore, unsurprisingly, we see that their freezing levels really go up as soon as they start to feel these shocks. The interesting question is, what would the observer do? Nothing actually happens to the observer. But when we look at their behavior, we see that they start to freeze as well, as if they had received some of these shocks. Now, of course, foot shocks are very artificial stimulus for rodents. So we figured what would happen if the observer knew what foot shocks felt like. So in other experiments, we kind of pre-expose the observer to a couple of foot shocks in a different environment. This can be done even a month before the actual observation. And then we perform this observation experiment. And when we do that, we now see that the observer has a dramatic freezing response, really as strong as that of the demonstrator. Now, with these experiments, what we have now is a phenomenon where the emotional distress of one rodent is basically shared by another rodent, which is a nice model of what I was calling emotional contagion before. Something that we observe, though, is that it's not just a one-way street. So if we look at what actually happens to the demonstrators, we see that demonstrators that are paired with an observer that doesn't react a lot to their distress show a lower level of freezing than demonstrators that are paired with an observer that also gets really scared when they see the phenomenon. So this has shown us that we basically, with this very simple paradigm, are looking at a combination of two effects. We have emotional contagion, where distress goes from the demonstrator to the observer, and we also have a certain degree of social buffering, where a calm bystander can help the demonstrator actually relax a little bit in this phenomenon. Now you might ask me, okay, you start with our experience in the Syrian boy, and now you show me rodents that are being scared of another one getting foot shocks. Do you think that the rodent actually feels the same thing that we would feel in that situation? Now that's obviously a really interesting question, but one that's also very difficult to answer. As neuroscientists, though, there's one thing we can do is ask whether the brain activity is similar in the rodent and in the human. Because if we're not dualist, if we see similar brain activity, there might actually be a similar experience behind it. And so now something that we are now doing more in the lab is using ultrasound imaging as a method that we can use in rodents that is also dependent on the hemodynamic response function that gives us images that we can directly compare to your human neuroimaging that we do in the lab. And I've shown you that in humans, when you see the pain of others, for instance, the cingulate is a really central brain region. So what we did was we imaged a slice of the rodent brain, including the cingulate, both while the rodents observe another one getting a shock and while they get a shock themselves. And when you overlap them, what you see is that the exact brain region that is so salient in humans when they see the pain of others also shows a similar effect in the rodent. So that is the beginning, let's say, of seeing that there are homologous brain regions in the two species that are about 100 million years apart in evolution, responding to witnessing the distress of others. So now we can do more interesting neuroscience. For instance, ask whether there is causality between activating your cingulate and sharing the distress of others. So what we do here is we take area 24, which is the homologue of the region we were seeing in the humans, and we can simply inject something like muscimol in there and see whether it would impair emotional contagion. So we perform our emotional contagion experiment either after we inject saline, and here we see that the observer shows this very high level of freezing that we had seen before. And this is how much they freeze if you interfere with the cingulate, showing that there is really an impairment of this emotional contagion. But you might figure, what have we impaired? Is it the ability to share the emotions of others, or is it just the ability to be scared at all? So what we can do to control for that is we can simply fear condition the rodent. So there during pre-exposure, while they experience these food shocks for the first time, we always play a beep tone before they get the shock. If you do that for a couple of times and then play back just a beep tone, what you see is also about 80% freezing. So that's a good control. But now deactivating the cingulate does not impair the freezing. So it's not like we have impaired the ability to be scared from a sensory signal, but we have impaired something that seems to be linked more specifically to being scared because of a social signal that comes from the distress of another animal. Now, what was interesting is that although we only injected the observer, we saw that it also slightly reduced the amount of freezing in the demonstrator that were getting the shocks, again, providing some evidence for this two-way communication between the two animals. Now, of course, another question we have is it actually really your pain neurons in the cingulate that get activated when you observe the pain of others? So in the rodents, we can look at that. So we implanted silicon probes in the cingulate region, and we were looking for neurons that are active when the animal itself feels pain. So now here you see the population activity of these neurons that respond when the animal itself feels a heat laser on their paw that creates a kind of burning sensation. If we dial down the heat laser to the point where they just feel something, but it's not painful, these neurons are not active. Now, what we can do is record from these neurons while the animal is witnessing someone else getting foot shocks. And this is the response when they see someone else getting a strong foot shock. This is when they get a slightly weaker foot shock. And this is when you just shock an empty grid. So, what you see here is a quantitative mapping of how much pain someone else is in onto neurons that respond to the animal's own pain, really showing at the single cell level what we were observing kind of decades before in the motor system of the monkey. And again, if we just present a fear conditioned tone, there's no response here. And I think that is really important because the cingulate is part of what in humans is now more and more called the saliency network. So it's important to have a salient control that these neurons do not respond to. But that brings us to ask ourselves, why would animals actually care about the emotions of others? Now, in humans, we have a tendency to look at empathy as being this warm, fluffy feeling that makes us nice people towards others. And a lot of people think that it might be really an extension of the warmth and care we have for our offsprings to other conspecifics. But I think there might be a much simpler explanation. So I live in Amsterdam, where there's mice pretty much in every house. So often at night, I have little mice roaming around in my kitchen. And I also have a cat, so sometimes the cat goes into the kitchen. And now these two different mice are in a rather different situation. One of them can directly see the cat enter the room and will, of course, start freezing, which is highly adaptive because if they stop moving, they stop making noise, and they usually that saves their life. Now, what about the other guy? They can't see in the cat directly, so what would they do? If they just keep roaming around, they'll end up making enough noise to end up as dinner. But if they experience emotional contagion, they will start to freeze as well, not because they directly saw the danger, but because they kind of felt it by using others as a fear antenna. And of course, the nice thing is if they don't just copy the behavior, but the actual emotion, they also prepare all of the other good stuff that happens when we're scared. So they get the whole activation of the fight and flight system involving hormonal responses and so on that prepares them to deal for this socially sensed danger. Now, if the cat gets bored and walks away, one of them sees it, can relax. And now the social buffering that we've also shown helps the other mouse realize that danger has gone, relax, and they can go on and steal some of our food. Now, I think what that shows is that sharing the emotions of others doesn't just have an altruistic purpose of being a nice person, but it can actually serve your own selfish purpose of adapting to the level of danger in the environment. And I think that this selfish purpose helps us better understand why the very same brain regions have evolved over hundreds of millions of years to share the emotions of others because it has this strong selfish purpose. Now, you might wonder if that's the case, then it's really important to learn basically where danger is and where danger is not. So, now some of the experiments we and others have done is, for instance, to put two mice or rats together in one environment, nothing bad happens. Then we put them in another environment where one of them gets a shock. Then the next day, we place them alone again in the two environments. And what we see is that both mice and rats will the next day freeze more in the environment in which they've seen bad things happen to others. So you might wonder where in the brain does the animal actually link the where with the what happened to others? And I think it's a safe bet to look, for instance, in the hippocampus for that, because of course, if you record from the hippocampus, you would see that individual place cells have a different response already at baseline in the two environments. So here you see cell one that has a strong response in what will become the safe environment. Cell two, for instance, has a strong response here in what will later become the shock environment. But then what happens while they witness shock to others? Well, what we see there is that a significant proportion of the dorsal hippocampus neurons start to fire whenever they see shocks being administered to the other animal. So now the question is, do these neurons actually link the where with the what happens to others? And we might expect that one way in which they could link the two things is that perhaps they could fire more if they are in the actual place in the field of that cell at the moment where they observe the shock. And that's exactly what we see. So the trick here is that if this is the part of the environment that the observer can explore, this is where the demonstrator is going to get some shocks. What happens is that the observer is not always in the same place in this environment while they observe the shock. So sometimes they're up here while they observe the shocks, then they move a bit to the back, and so on. Now, this is the field of one of the place cells in the hippocampus, and therefore sometimes the animal is currently within the place field of that cell where they observe the shock, and sometimes not. So here is a trial where they were out of the place field, and what we see here in this graph is so they're low in the place field, and when they see the shock, there's a very small response to one spike here. Then on another trial, the animal is actually spot pretty close to the maximum firing of the place field of this cell. And now we see that there is a much stronger response for this cell when it sees the shock of others. If we do that not just for one cell, but for all the cells that we record, we see that there is a general trend where basically these cells respond more strongly if the observer was in the place field of that neuron when they see the shock than when they're not. So this seems to be one of the ways in which the brain can really map danger perceived through others onto a particular location in space. And that then really allows you to adapt your foraging to the amount of danger that your friends have encountered there. But now the other question we had when we looked at the Syrian boy is how the sharing of emotions might actually motivate you to help others or not to do things at least that harm others. But what I've shown you so far is basically that if I see the Syrian boy, the first thing I should do is learn that it's perhaps not a good idea to go to Syria at a certain moment in time because bad things happen there. Now, would there be more to it? Would you actually be motivated to help? So what we did there is we've done some human experiments, but I'm going to present one of our rodent experiments. What we've done is we've given the rat two levers. When they try the first lever, they see, gee, this one is easy to press, that's nice, and I get a nice reward. Then they try the other one, they realize, gee, this one is twice as hard to press, and actually get the exact same reward. So that's not my optimal strategy. Most of them will tend to develop a strong preference for the easy lever. And as soon as they do that, we play a trick on them. So now each time they use their favorite lever, we give a little foot shock to the guy next door. So now the question is, if a rat doesn't like to cause pain to others, we should see them shift to the other lever because they can still get food. They have to work a little harder, but they can prevent pain. And that's exactly what we see. So, as a population, both males and female rats will move away from the shock lever as soon as it causes harm to others. So, this is true for men and females. So, the stereotype that we men don't care is untrue. But something that we observed is that both in the males and in the females, there is actually tremendous individual variability. So some of them completely stop to use that lever as soon as they see shocks, and others don't care at all. The Trumps in here that will just continue to use a profitable strategy, even if they realize that it hurts others. And this is something that we'd be really interested in understanding better. But so far, we have not identified the real differences in the brain between these animals. But what we can now do is a very simple experiment. We can figure it. So we knew that the cingulate was really important for the emotional contagion. Would it also interfere with the animal's willingness to shift away from the shock lever? So we again just inject muscimol in there. What we see is that if we inject saline, they move away from the shock lever. If we inject muscimol in the cingulate, they don't. So this is really one, I think, of the few causal evidences we really have to show that substrates that allow you to share the distress of others actually have a real impact on your social decision making in terms of avoiding harm to others. Now, I'd like to conclude by asking a little bit. So, we've seen that rodents and humans seem to share a machinery to share the emotions of others. So, that shows that to a certain degree, it's biologically pre wired. But does that mean that it just happens to us, or is it something that we can control? Now, we came to that question actually from looking at what happens in psychopathic criminals. So, what we did was we went to high security prisons and recruited psychopathic criminals. They had done really pretty terrible things on violent rape or sometimes murder. And we were wondering, would this show an abnormal sharing of the emotions of others? And is that why they don't care so much about what they do to others? Now, the experimental design was pretty simple. We first let them view others experiencing pain. Then the fun part of the experiment started, where we went into the scanner room and hit these terrible criminals on their hands to teach them a lesson. So the second part was aimed at testing whether perhaps they just don't feel pain, be it their own or those of others. And the first part, of course, to measure the empathy. Now, when we were looking at what happens while they were experiencing pain themselves, what we saw was their response was extremely similar to that of the healthy volunteers and involved all the brain regions you would expect, kind of the cingulate, the insula, sensory, motor cortices. Differences, though, appeared where they were simply watching the pain of others. The controls had strong activity, and the psychopathic criminals had much lower activity. And if we focused onto the brain regions involved in their own pain, we observed reduced brain activity in all of these shared circuits, including the ones for sharing actions, sensations, and emotions. So you might figure, yes, indeed, they seem to have a broken kind of mirror system. They're just incapable of empathy. But when you spoke to their victims, what you see is that they were often surprised because they figured at first contact, and he was very nice, seemed to be really feeling what was going on in me. But then suddenly they switch into this cold mode where they just start to do whatever they want with me. So we figured, what would happen if we asked them to try and empathize with the victim? So we showed them the movies again, but this time we told them to really try to empathize with the victim in the movies. And what we saw was that that completely abolished the brain differences. So now the psychopathic criminal had a pretty normal recruitment of their own emotion while they witnessed the emotion of others. So what that showed us is that psychopathy is not so much the absence of empathy, but a tendency not to use the empathy spontaneously. And then they themselves sometimes see that as a talent, is the fact that for them, empathy doesn't get in the way of achieving their goals. And so these were criminals, but I think we all have met some very effective managers or bankers that seem to share this talent and not to have empathy get in the way of their decision making. But more generally, it alerted us to the fact that sometimes you can have an ability to empathize without using it all of the time. Now, over the years, we've seen many cases in which most of us actually regulate our empathy. One case that has been documented by other groups is the in group out group difference. So, there's a famous experiment in which Tanya Zinger, for instance, showed people the pain of someone and told them, and by the way, he likes the same football team that you like. And you had a lot of activity in the pain system. And then she showed them sometimes pain and said, well, this one actually likes this other football team that you don't like so much. And then the activity went down. The same has been shown for differences in race. We've shown a couple of experiments in which you either see pain of which you know that you caused it because you took a certain decision or made a mistake. And we can measure much higher activity in the pain system than when you see the same pain, but you didn't cause it. The person, for instance, themselves caused it. And finally, we also just showed people emotional Hollywood movies and asked them to either empathize with the main character or to take a more detached view. And simply by changing the instruction, people could up and down regulate significantly the brain activity in these systems. So, with all of that together, I think what we're starting to see is that Empathy, yes, it is something that we share with mice and rats that is kind of pre-wired by evolution, but it is also something that we can very effectively up and down regulate at will to kind of fit the kind of situation we're in currently. And now to conclude, let me try to put all of that together into the brain. So we started by asking a bit, why is it that we don't just understand what happens in this boy, but get moved so much by him? And I think what I've shown you is that this might be because we don't just activate our visual cortices when we see him, but when we see his actions, we recruit our own motor system as if we'd be performing similar actions. While we observe his sensations, we activate our somatosensory system as if we'd have similar sensations. And what I've showed you in the greatest detail is that when you witness the emotion of the other, you recruit cells and brain regions involved in your own emotions as if you were in his stead. And with all of that recruited, of course, you stop to just understand. Because in a way, your brain makes you be this boy while you observe him. You really can feel his emotions. And I hope that by combining human and rodent experiments, we can go beyond what we can just do with one of the species by being able to as well do causal experiments in which we could show that preventing this activity prevents the sharing of the emotion and also your tendency to go out and avoid actions that harm others. And we could also peek into these blobs and show that it is really the same neurons involved in your own pain that get reactivated. And also that there is actually this really long evolutionary history to sharing the emotions of others. And finally, I hope to have shown you as well that although this might be biologically pre-wired, both the situation and your own personal decisions can influence how much you will engage the system and therefore share the emotions of others and care to help them. And finally, I'd like to thank Valeria, my wife, with whom I co-lead the lab. We design all experiments together, and it's really an amazing experience to be able to spend 24 hours a day with your most loved person. But I'd like to thank all the amazing people we have in the lab to combine the human and the rodent experiment, and a lot of really amazing alumni whose work I've featured here. And finally, I'd like to thank NeuroPSI and the Chen Foundation for getting us together today. Thank you for your attention.